Hello there, and welcome to the Sleepy Science Channel. Tonight, we are going to drift together into one of the most quietly astonishing places in the universe. And you don't have to go anywhere to find it. It is already inside you. Right now, as you settle in and let your body grow still, every single cell in your body is humming softly with a molecule so thin it cannot be seen, so long it could stretch to the moon and back thousands of times, and so wise it has been carrying the story of life on Earth for nearly 4 billion years. It is called DNA. And tonight, you're going to hear its secrets, not as a science lesson, not as something to memorize or understand all at once, but as a slow, gentle unfolding, like a letter written in a language older than words, read to you one quiet page at a time. Let yourself breathe. Let your shoulders drop. This is a story written for you, inside you, and it has been waiting a very long time to be told. Somewhere deep inside you, tucked inside the nucleus of nearly every cell in your body, there is a molecule that holds everything, not in a dramatic way, not the way a treasure chest holds gold, more the way a quiet river holds the memory of rain. It simply carries it faithfully through time. DNA is short for deoxyribonucleic acid, but you never have to remember that. What matters is the feeling of it, a long spiral ladder twisting gently, like something that decided gravity was optional. Scientists call this shape the double helix, and if you close your eyes and picture two ribbons winding softly around each other, rising upward without hurry, you already have the right image. It is one of the most beautiful structures in all of nature, and it fits inside a space you could not see with the naked eye. Each rung of this spiral ladder is made of just 4 chemical letters: A, T, C, and G. That's all. 4 letters, the way music is built from just a handful of notes. And yet, from those 4 letters, the instructions for every living thing on Earth have been written. Every tree, every bird, every sleeping dog, every person reading words on a screen. All of it spelled out in combinations of those same quiet 4. If you were to take just one cell from your body, one tiny, invisible cell, and carefully pull out all the DNA coiled inside it, that single thread would stretch to about 2 meters long. 2 meters from a cell you cannot see. The body has a remarkable gift for folding things gently, patiently, into spaces that seem far too small. And you have a lot of cells. Somewhere around 37 trillion of them, give or take. If you were to lay all the DNA from all your cells end to end, the thread would stretch from here to the sun and back, not once, but more than 300 times. You are, quietly and without fuss, made of something almost incomprehensibly vast. The information stored in your DNA, the full set of instructions for making and running a human body, is called your genome. And if you were to print it out, letter by letter, it would fill about 3,000 books, each one 500 pages long. You are carrying a library inside you, and it never makes a sound. Here is something that might make you smile softly in the dark. Your genome and the genome of any other person on Earth are about 99.9% identical. That small remaining fraction, just 0.1%, is what makes each of us different. Our eye color, the exact shape of our nose, tiny variations in how our bodies work. Everything that makes you distinctly you is written in a fraction the size of a whisper, and yet That whisper matters. There are 3 billion letters in the human genome, and 0.1% of 3 billion is still 3 million differences. 3 million quiet variations sitting in your cells right now, making you exactly and only yourself. DNA does not just sit there, though. It is constantly being read. Right now, as your breathing slows and the room grows quieter around you, tiny molecular machines inside your cells are moving along those long DNA threads, like slow readers running their fingers across a page, finding the sections they need, reading the instructions, and carrying them out. It happens all the time, without your awareness, without any effort from you at all. These sections of DNA that get read, the ones that carry actual instructions for making proteins, the building blocks of everything your body does, are called genes. You have somewhere around 20,000 to 25,000 genes. That number surprised scientists when they first counted them, because it is not as many as they expected for something as complicated as a human being. A water flea has more genes than you do. Nature, it turns out, is less about quantity and more about how gently and cleverly things are arranged. What fills the rest of your DNA, the parts that are not genes, was once called junk DNA because scientists were not sure what it did. That name has softened considerably over the years. Much of it turns out to be something like a vast and subtle control system full of switches and signals that decide when genes get turned on and when they stay quiet. It is not junk at all. It is more like the spaces between musical notes, essential to the rhythm, invisible to anyone who is only listening for the loud parts. Genes are inherited, passed down from parents to children, generation after generation, in a long, slow river of information. You received one copy of each chromosome from your mother and one from your father, and so the instructions inside you are a gentle blend, a quiet conversation between two separate histories, braided together the moment you began. Chromosomes are the chapters that DNA is organized into. Humans have 46 of them, 23 pairs. Each one a tightly wound bundle of DNA, coiled and folded with extraordinary care. If you imagine the full 2 meters of DNA in each cell as a very long piece of thread, the chromosomes are what you get when you wind that thread around tiny spools, then arrange those spools neatly on a shelf. The body is very tidy about this. It has to be. 2 of those 46 chromosomes are the sex chromosomes, the ones labeled X and Y. People with 2 X chromosomes typically develop differently from people with 1 X and 1 Y, though the full story of how biological sex develops is far more nuanced and wonderful than a simple letter can capture. Nature, as always, prefers complexity to simple answers. The very tips of your chromosomes are capped with something called telomeres, little protective sequences of DNA that repeat the same short phrase over and over, like a sentence written at the end of a document just to protect the important words from fraying at the edge. Telomeres shorten a little each time a cell divides, and scientists study them closely as one of the quieter clocks of biological aging. They are, in a sense, a soft and patient record of time. And if you are feeling sleepy right now, if your eyes are growing heavy and the world is becoming softer around you, that is a perfectly good moment to let your attention drift a little. You do not need to hold on to every word. The facts will find you again when you need them. For now, just feel the quiet. If you are enjoying this gentle journey through the world inside your cells, consider leaving a comment below. We love hearing which facts made you pause and smile. And if you haven't already, there is a whole playlist waiting for you full of nights just like this one. Your DNA has been with you from the very beginning, from the moment a single cell divided to become two, and two became four, and so on, in a slow and patient blooming. Every division carried your DNA along, copied faithfully, like a song passed carefully from one musician to the next. The copying is done by a molecule called DNA polymerase, and it works with remarkable accuracy, making on average only about one mistake per billion letters copied. If you imagine typing out the entire works of Shakespeare, roughly 5 million characters, with that level of accuracy, you would expect only a tiny fraction of a single mistyped letter. The story of DNA did not begin with humans, or even with animals. It reaches back in an almost dreamlike way to the very earliest life on Earth. Single-celled organisms drifting in ancient oceans, simpler than anything alive today, but already carrying the essential secret. Instructions written in a chemical alphabet, passed from one generation to the next, changing slowly over time. The 4 letters of DNA, A, T, C, and G, stand for 4 molecules called nucleotide bases: adenine, thymine, cytosine, and guanine. You do not need to remember these names. What is lovely about them is the way they pair. O always bonds with T, and C always bonds with G, always, without exception. This is called base pairing, and it is what makes DNA so beautifully reliable. Each strand of the double helix is a perfect mirror of the other because of these faithful pairings. This complementary structure is also what makes copying DNA possible. When a cell prepares to divide, the 2 strands of the double helix gently unzip down the middle, the way a zipper opens, slowly and without urgency. And each half becomes a template for a new partner strand. 2 complete DNA molecules emerge from one, each identical to the original. It is a kind of quiet, cellular magic. The unzipping is carried out by a molecule called helicase. It moves along the DNA, unwinding the helix ahead of it, and the name is rather poetic. It comes from the Greek word for the sun, helios, because of the spiral shape, a molecule named for sunlight, working in the deep, dark interior of your cells. Once the strands are separated, another molecule, the aforementioned DNA polymerase, moves along each template strand, reading the exposed letters and attaching the correct matching bases one by one, building the new strand as it goes. It moves at a speed of about 1,000 letters per second in human cells. Quietly, quickly, perfectly, and you never notice it at all. What happens when a mistake does slip through? The body has editors. Several different repair systems constantly scan DNA, looking for errors, mismatches, or damage, and correcting them before they cause problems. One of these systems is called mismatch repair. Another is called nucleotide excision repair. Names that sound complicated, but describe something almost tenderly careful. The body reading its own text, finding the small errors, and gently fixing them. DNA can also be damaged by outside forces, ultraviolet light from the sun, certain chemicals, or simply the ordinary chemistry of living. When this happens, repair systems are activated almost immediately. The cell pauses what it is doing, attends to the damage, and then continues. It is a remarkably patient process, and it happens countless times in your body every single day. The information in DNA does not go directly to where it is needed. It travels in a kind of molecular relay. First, a section of DNA is copied into a related molecule called RNA, ribonucleic acid, in a process called transcription. RNA is a single-stranded cousin of DNA, and it carries the message out of the nucleus and into the wider cell where the actual work happens. Once outside the nucleus, the RNA message, now called messenger RNA or mRNA, is read by a molecular machine called a ribosome. The ribosome moves along the mRNA, reading 3 letters at a time, and for each triplet, it attaches a specific amino acid. Amino acids are the building blocks of proteins, and as the ribosome reads further along the message, the chain of amino acids grows longer. Eventually, when the ribosome reaches a stop signal, the chain is released, and it folds spontaneously, elegantly, into the 3-dimensional shape of a protein. The genetic code, the dictionary that translates triplets of DNA letters into amino acids, is shared by almost every living thing on Earth. The same codon that tells a bacterium to add the amino acid leucine tells your cells the same thing. This universality is one of the quietest and most profound pieces of evidence that all life on Earth shares a single ancient origin, a whispered connection running through billions of years and across every living thing you have ever seen. Proteins do nearly everything. They carry oxygen through the blood, speed up chemical reactions, build structures, send signals, defend against infection, and regulate the activity of genes themselves. Your body makes somewhere around 100,000 different proteins, all of them built from combinations of just 20 amino acids, all of them ultimately encoded in those 3 billion letters of DNA. The idea that DNA holds the master instructions for life was understood in a general way for many years before the details were known. It was in 1953 that James Watson and Francis Crick, working with X-ray images created by Rosalind Franklin and Raymond Gosling, described the double helix structure. Franklin's contribution, made through painstaking experimental work, was essential. Her X-ray photograph of DNA, known as Photo 51, revealed the helical shape with striking clarity. Take a slow breath in, let it out. Notice how effortlessly your body knows what to do. That ease, that quiet, unthinking competence, is DNA at work. If something about these stories is helping you feel peaceful tonight, please leave a comment. It truly means a great deal. And if you want more nights like this one, our playlist is always there, waiting like a lamp left on in a quiet room. Your DNA is not just a set of instructions, it is also a kind of history book. Written into the sequence of your genome are the records of everything your ancestors survived, every environment they adapted to, every ancient moment that shaped the lineage that eventually produced you. Evolution works by introducing variation into DNA, small copying errors, rearrangements, insertions, deletions, and then allowing the environment to act as a quiet filter, favoring the variations that help organisms survive and reproduce. Over millions of years, this slow and patient process has shaped every living thing on Earth, writing new chapters into genomes without ever picking up a pen. One of the most illuminating discoveries in modern genetics is how much human DNA we share with other species. You share about 98.7% of your DNA with chimpanzees, our closest living relatives. The difference of about 1.3% represents millions of individual changes accumulated over roughly 6 million years of separate evolution. But the similarities go further and become more surprising the deeper you look. You share about 85% of your protein-coding genes with mice, about 60% with fruit flies, about 31% with yeast, single-celled fungi that have been making bread rise for thousands of years. The genes involved in basic cellular functions, dividing, repairing, energy metabolism, are some of the oldest and most conserved in all of life, because they have been doing their jobs well for so long that changing them usually causes more harm than good. You even share some DNA with bananas. About 60% of human genes have a recognizable equivalent in bananas. This is because the basic toolkit of life, the genes that manage cell division, energy production, and DNA repair, was worked out so early in evolution that it has been largely preserved ever since. These ancient conserved genes are called housekeeping genes because they manage the everyday functions that every cell needs to stay alive. They are the background hum of life, constant and reliable, the same quiet melody playing in cells from mushrooms to elephants to the sleeping person in the bed next to you. There are also stretches of DNA in your genome that came long ago from viruses. About 8% of the human genome consists of sequences derived from ancient viral infections, viruses that inserted their DNA into the genomes of our ancestors, sometimes millions of years ago, and whose genetic remnants were passed down through the generations ever since. Some of these sequences have been quietly repurposed by evolution and now perform useful functions. Others simply sit there, passengers in the genome, carried forward through time without any particular purpose, like dried flowers pressed between the pages of a very old book. One of the most extraordinary examples of repurposed viral DNA involves the placenta. Proteins called syncytins, which are essential for the formation of the placenta in mammals, appear to have originated from viral envelope proteins, the proteins that ancient viruses used to fuse with host cells. Evolution, remarkably, took the machinery of infection and turned it into something nurturing. Within the human genome, there are also large regions of repetitive sequences, the same short phrase of DNA letters repeated hundreds or thousands of times in a row. These repetitive elements, with names like ALU elements and line 1 elements, make up a large portion of the genome. They're mostly silent now, but they represent echoes of ancient mobile DNA, sequences that once could copy themselves and insert elsewhere in the genome, like slow-moving molecular passengers that eventually settled down. The study of how genes are switched on and off, turned active or silenced, is called epigenetics, and it is one of the most gently fascinating frontiers of modern biology. Your DNA sequence itself rarely changes during your lifetime, but which parts of it are actively being read can shift in response to your environment, your experiences, even the seasons. The same genetic text can be highlighted differently in different cells, and even in the same cell at different times. Epigenetic changes are made through chemical modifications to the DNA itself or to the proteins it is wrapped around. One common modification is called methylation, a tiny chemical tag attached to certain DNA letters that generally quiets the activity of nearby genes. Another involves modifications to the histone proteins around which DNA is coiled, loosening or tightening the coils to make genes more or less accessible to be read. These modifications can, in some cases, be influenced by experience. This is a field still being actively explored, and it carries a certain quiet wonder. The idea that the life you live leaves gentle marks on the molecules inside you, changing the way your genome is read. Not the text, but the reading of it. Some epigenetic changes can even be passed on to the next generation, though this is still a topic of active research and careful nuance. The idea that something of what a parent experienced might echo softly in the biology of their children is one of the more quietly remarkable possibilities of modern science. If you are still awake and your mind is drifting pleasantly through these ideas, that is exactly right. There is nowhere you need to be, nothing you need to hold, just the soft company of these ideas and the quiet. There is a part of every cell's life that is especially orderly and careful. The moment when a cell decides to divide. Before it does, it must copy all of its DNA, all 3 billion letters, and distribute one complete copy to each new daughter cell. This process, called mitosis for ordinary cell division and meiosis for the formation of reproductive cells, is choreographed with extraordinary precision. During meiosis, the special cell division that produces eggs and sperm, something remarkable happens. The chromosomes from your mother's side and your father's side come together in pairs, and they exchange segments with each other. This process is called recombination or crossing over, and it is the reason why you are genetically unique. The chromosomes you pass on to any children you might have will be shuffled versions of the ones you received, a new combination, never seen before, never to be seen again. Recombination happens at specific locations along the chromosomes, though there is some randomness to exactly where. On average, each chromosome undergoes 1 to 3 crossover events during meiosis. The result is a reshuffling of genetic material that has been happening in every sexually reproducing species for hundreds of millions of years. One of evolution's most elegant tools for generating variety. Your genetic uniqueness begins at the moment of fertilization. One sperm, chosen from among hundreds of millions, meets one egg, and the 2 sets of chromosomes, 23 from each parent, come together. Given the number of possible chromosome combinations from just the meiotic shuffling, plus the variation introduced by recombination, the probability of any specific person existing is almost incomprehensibly small. And yet, here you are. Each of your cells carries a complete copy of your genome, the full 3 billion letters. But different cells read different sections of it, which is how a liver cell and a neuron and a skin cell can be so different from one another despite containing identical DNA. The process that determines which genes are active in which cells, cell differentiation, is one of the central mysteries and wonders of developmental biology. During development, a single fertilized cell divides and divides, and as it does, each new cell receives signals that gradually narrow its identity, turning some genes on, silencing others, until each cell knows what it is meant to become. The signals come from neighboring cells, from chemical gradients flowing across developing tissue, from the cell's position in the growing body. It is a process of exquisite coordination, unfolding without any central director, guided only by local conversations between cells. The genome you carry today is not perfectly identical to the one you were born with. Over a lifetime of cell divisions, occasional errors accumulate, single letter changes, small deletions, tiny insertions, scattered across the genomes of different cells in your body. This means that, in a sense, your body is a collection of slightly different genomes, a mosaic of related but not quite identical genetic texts. Most of these changes have no effect at all. They occur in parts of the genome that are not critically important, or they are repaired, or the cell that carries them quietly reaches the end of its life and is replaced. Cells do not live forever, and they are not meant to. Most cells in the body have a limited lifespan, after which they undergo a carefully orchestrated process of self-dismantling called apoptosis, programmed cell death. It sounds melancholy, but it is actually a sign of health. Apoptosis is the body's way of removing cells that are old, damaged, or potentially dangerous, maintaining the overall integrity and health of the tissue. About 50 to 70 billion cells in your body undergo apoptosis every single day, quietly and without fuss. The cells of the lining of your small intestine are replaced every 2 to 4 days. Skin cells live for a few weeks. Red blood cells circulate for about 4 months before being retired. Some cells, like neurons in certain parts of the brain, stay with you for a lifetime. The cells in your cerebral cortex are almost exactly as old as you are. The neurons you used to read your first book are most likely still there. There are some cells that divide so slowly they seem almost timeless. The beta cells of the pancreas, which produce insulin, are replaced only rarely. Some of them may persist for decades. The cells of the heart muscle divide very slowly throughout life, meaning the heart you have now still contains many of the original muscle cells from when you were a child. All of this, every division, every repair, every quiet act of maintenance, is coordinated by the instructions in your DNA, not by any single molecule acting alone, but by thousands of molecules in constant conversation, reading and responding to each other in a language that took billions of years to develop. If the quiet wonder of all this is helping you settle into sleep, please remember to like this video. It is such a simple thing, but it helps more than you might imagine. And our playlist of gentle science stories is there whenever you need a soft place to land at the end of the day. One of the most quietly revolutionary moments in the history of science came in 2003, when the Human Genome Project was declared complete. Scientists from countries around the world had spent 13 years painstakingly reading out all 3 billion letters of the human genome, mapping, sequencing, and compiling the full set of human genetic instructions. It was the longest book ever read, and the reading of it changed everything. The project was not the work of a single genius in a laboratory. It was a collaboration, an extraordinary international effort involving thousands of scientists, technicians, and support staff across 20 countries. The data was made freely available to anyone in the world, placed in public databases where researchers could access it without charge or restriction. It was a gift from science to the future. Even before the Human Genome Project was finished, a private company called Celera Genomics, led by Craig Venter, had begun its own genome sequencing effort using a different and faster approach. The competition between the public project and the private effort accelerated the timeline considerably. Sometimes, the most productive scientific moments come from the creative tension between different approaches reaching for the same goal. The genome that was published in 2003 was actually a composite assembled from the DNA of several anonymous donors, and it was not quite complete. Certain regions, particularly around the centromeres and telomeres and other highly repetitive sections, resisted sequencing with the technology available at the time. Those gaps persisted for nearly 20 years. It was not until 2022 that the first truly complete human genome sequence was published. Every single letter from one end of every chromosome to the other. This was made possible by new sequencing technologies that could read very long stretches of DNA at once, rather than having to assemble thousands of short fragments. The complete sequence added about 200 million new letters to what was previously known, revealing new genes and regulatory elements hidden in regions that had been inaccessible before. The technologies used to read DNA have changed remarkably over the decades. The first method, developed by Frederick Sanger in the 1970s, was elegant but slow. It could read perhaps 100 to 1,000 letters at a time and required considerable expertise. Sanger sequencing earned its inventor a Nobel Prize and laid the foundation for everything that came after. Modern sequencing technologies can read billions of letters in a single run in a matter of hours at a cost that has dropped by several orders of magnitude over the past 2 decades. Sequencing a human genome cost about $100 million in 2001. By the early 2020s, the cost had fallen to somewhere in the range of $1,000, and it continues to drop. What once required the resources of an international scientific consortium can now be done in a small laboratory. This has opened up remarkable possibilities. Doctors can sequence the genome of a patient's tumor to understand exactly which mutations are driving its growth and which treatments might work best. Researchers can sequence thousands of genomes from people with a particular disease, comparing them to thousands of genomes from healthy people to identify the genetic variants that increase risk. Archaeologists can sequence DNA extracted from ancient bones, reading the genomes of people who lived tens of thousands of years ago. Ancient DNA is a particular kind of wonder. DNA degrades over time. It breaks into smaller and smaller fragments, and chemical modifications accumulate. But in the right conditions, cold, dry environments like permafrost or caves, DNA can survive for astonishing lengths of time. The oldest DNA ever sequenced came from a mammoth frozen in Siberian permafrost for about 1 million years. From ancient human remains, scientists have learned things about human prehistory that could not have been known from archaeology alone. They have traced the roots of ancient migrations, identified previously unknown groups of ancient humans, and discovered that modern humans interbred with Neanderthals and Denisovans, 2 other human species now extinct, before those groups disappeared. Most people today carry a small percentage of Neanderthal DNA, a quiet echo of encounters that happened tens of thousands of years ago, still present in every cell of your body. Let that rest with you gently for a moment. Somewhere in your genome, there may be a whisper of something ancient, carried forward from a time when the world was wilder and colder across thousands of generations, all the way to you, reading or listening to this, warm and still in the quiet of tonight. The genome does not just tell a story about what you are. It tells a story about where you came from, and not just you personally, but all of us together as a species. Every person alive today is descended in an unbroken line from a relatively small group of humans who lived in Africa somewhere between 50,000 and 200,000 years ago. The genetic evidence for this is written clearly in the patterns of human genetic diversity around the world. Diversity that is greatest in Africa and decreases as you move further away, reflecting the successive founder populations that moved into new territories, each carrying only a subset of the diversity present in the group they came from. There is a concept called mitochondrial Eve, the most recent common matrilineal ancestor of all living humans. She was a real woman who lived in Africa, probably more than 100,000 years ago. She is called Eve, not because she was the only woman alive at the time, but because all mitochondrial lineages from other women of her era eventually died out over generations. Her mitochondrial DNA, passed from mother to children in an unbroken line, is the ancestor of all mitochondrial DNA alive in the world today. Mitochondria are tiny structures within cells that generate energy, often called the powerhouses of the cell. They carry their own small circular genome, separate from the nuclear genome, because they were once free-living bacteria that were engulfed by another cell roughly 2 billion years ago and never left. The relationship became permanent and mutually beneficial, a partnership so successful that all complex life on Earth is built upon it. The mitochondrial genome is small, only about 16,000 letters compared to the 3 billion in the nuclear genome, but it is passed down exclusively through the maternal line because eggs contain mitochondria and sperm do not, or their mitochondria are typically destroyed after fertilization. This makes mitochondrial DNA an extraordinarily useful tool for tracing ancestry through the female line. There is also a concept called Y chromosomal atom, the most recent common patrilineal ancestor of all living men, traced through the Y chromosome, which passes from fathers to sons. Y chromosomal atom lived more recently than mitochondrial Eve, probably around 200,000 to 300,000 years ago, though estimates vary depending on the methods used. Neither Adam nor Eve were the only humans of their time. They were simply the ones whose particular lineage, in each respective case, has survived unbroken to the present. The vast majority of people who were alive alongside them left no surviving matrilineal or patrilineal descendants, not because anything happened to them, but because the unbroken chains of mother to daughter or father to son inheritance are easily broken over generations. Within the human genome, there are regions called SNPs, single nucleotide polymorphisms, which are positions where the DNA letter varies between individuals. A person might have an A at a certain position where most other people have a G. These small variations accumulate over generations and can be used to trace ancestry, estimate the time when different populations diverged, and understand the history of human migration. Direct-to-consumer genetic ancestry testing, which became widely available in the 2000s, uses patterns of SNPs to estimate a person's ancestral origins, comparing their genome to reference populations from different parts of the world. The results are probabilistic estimates, not precise genealogies, but they can offer a sense of the varied and often surprising geographic origins woven into any one person's heritage. Within families, DNA can be used to establish biological relationships with great precision. The probability that two random individuals share the same pattern of genetic markers across multiple locations in the genome is vanishingly small, which is why DNA evidence has become so important in contexts where establishing identity matters. Forensic genetics, the use of DNA in criminal justice, began in the 1980s. The first criminal conviction based on DNA evidence in the United States came in 1988. Since then, DNA analysis has both helped convict the guilty and, perhaps more importantly, exonerated people who were wrongly convicted, in some cases after they had spent many years in prison. Breathe gently now. Notice the weight of your body resting where it lies. You carry all of this history inside you, not as a burden, but as a kind of deep, quiet companionship. Billions of years of life finding its way to this moment. If you are finding peace in these stories, a like on this video helps us reach more people who might need a gentle place to rest. And there are many more stories waiting for you in our playlist. Whenever you are ready. The relationship between DNA and disease is one of the most actively studied areas in all of medicine, and it is also one of the most gently illuminating, because it shows just how much of health is written in the spaces between the expected and the actual. Some conditions are caused by mutations in a single gene, a single letter changed, a section missing, or a section duplicated in ways that disrupt the protein the gene encodes. These are called monogenic or single gene disorders, and there are thousands of them, though most are individually rare. One of the most studied is cystic fibrosis, caused by mutations in a gene called CFTR, which encodes a protein that regulates the movement of salt and water in and out of cells. Without a functioning version of this protein, mucus in the lungs and other organs becomes thick and sticky rather than thin and fluid. The mutations that cause cystic fibrosis are carried by many people with no symptoms. It is a recessive condition, meaning that two mutated copies are needed for the condition to manifest. Sickle cell disease is another well-known single-gene condition caused by a specific mutation in the gene for hemoglobin, the protein that carries oxygen in red blood cells. The mutation changes one amino acid in the hemoglobin protein, causing the protein to clump together under certain conditions and distort the shape of the red blood cells. The same mutation in single copy actually provides some protection against malaria, which is why it became common in populations from regions where malaria has historically been prevalent. Evolution does not optimize for comfort or ease. It simply rewards whatever helps organisms survive long enough to reproduce. Other conditions involve many genes, each contributing a small amount of the overall risk. These are called polygenic conditions. Heart disease, type 2 diabetes, many forms of depression, and most common cancers are polygenic, the result of many small genetic variations interacting with each other and with environment, diet, lifestyle, and chance. Understanding polygenic risk is one of the great ongoing projects of modern genetics. Cancer, in particular, is fundamentally a disease of DNA. Every cancer begins when a cell accumulates mutations that disrupt the normal controls on its growth, causing it to divide when it should not, to resist the signals that would normally trigger its death, to spread where it should not go. Different cancers have different characteristic mutations, and identifying those mutations in a patient's tumor can guide decisions about which treatments are most likely to help. The mutations that drive cancer are called driver mutations, the specific changes that give a cell a growth advantage. Other mutations that accumulate in cancer cells but do not themselves drive growth are called passenger mutations. A cancer genome might contain hundreds or thousands of mutations, but typically only a handful are doing the driving. Some inherited mutations increase the risk of certain cancers. The BRCA1 and BRCA2 genes, for example, encode proteins involved in repairing DNA damage. Inherited mutations in these genes can significantly increase the lifetime risk of breast and ovarian cancers. Many people now choose to be tested for these mutations so that they can make informed decisions about monitoring and preventive care. The idea of reading a person's genome to guide medical decisions, personalized medicine or precision medicine, has been a vision of medical researchers for decades, and it is gradually becoming a reality. Pharmacogenomics is the study of how genetic variations affect a person's response to medications. Some people metabolize certain drugs more quickly than others because of variations in the genes that encode drug metabolizing enzymes. And knowing this can help doctors prescribe the right dose from the start. Gene therapy, the idea of correcting faulty genes or adding new ones, has moved from science fiction to clinical reality in the past decade. Treatments using various gene therapy approaches have been approved for conditions ranging from certain inherited blindness disorders to rare metabolic diseases to some cancers. The field is still young, still cautious, still learning, but the progress has been remarkable. One of the most celebrated developments in gene editing is CRISPR-Cas9, a technology derived from a bacterial immune system that allows scientists to make precise changes to DNA sequences with unprecedented ease and accuracy. The name stands for Clustered Regularly Interspaced Short Palindromic Repeats, but what matters is the elegance of how it works. A short piece of RNA guides the molecular scissors to exactly the right location in the genome, and the cut triggers the cell's own repair machinery to fix or alter the sequence. Let that fact sit softly. Somewhere inside a bacterium, billions of years ago, a mechanism for recognizing and cutting the DNA of invading viruses was being quietly refined. Scientists discovered it, understood it, and adapted it into a tool for editing the genomes of any living thing with a precision and simplicity that seemed, not long ago, impossible. That is the kind of story that deserves a moment of quiet appreciation before sleep. The world of DNA is full of smaller mysteries, quieter wonders, the kind that do not make loud headlines, but settle into the mind like light through a window in the late afternoon. There is, for instance, the question of how the human body manages to fold 2 meters of DNA into a cell nucleus only about 6 micrometers across. This is not just a matter of cramming a lot of material into a small space. The folding is precise and meaningful. Genes that need to be active in a given cell are positioned near the center of the nucleus, where they are more accessible to the molecular machinery that reads them. Genes that are silenced tend to be tucked away near the nuclear periphery in regions of tightly compacted DNA. This three-dimensional organization of the genome inside the nucleus is itself a layer of gene regulation, a spatial language overlaid on top of the linear sequence of letters. Scientists have developed techniques for mapping this three-dimensional structure in different cell types, revealing a complex, dynamic architecture that changes as cells develop, divide, or respond to signals from their environment. The proteins that DNA is wrapped around are called histones, and they are not passive spools. They carry chemical modifications, tags added by specialized enzymes, that affect whether the DNA coiled around them is accessible or not. A histone with one set of modifications might keep the DNA tightly coiled and silent. A different set of modifications might loosen the coil and allow the DNA to be read. The language of histone modifications is called the histone code, and it is still being deciphered. Non-coding RNAs are another layer of complexity. RNA molecules that are transcribed from DNA but do not carry instructions for making proteins. Instead, they perform regulatory functions, helping to control which genes are active, guiding the modification of other RNA molecules, and participating in a vast and still being understood network of molecular communication. There are many types of non-coding RNAs, with names like microRNA, long non-coding RNA, and small nuclear RNA, a quiet vocabulary for a vast and largely invisible world. MicroRNAs are particularly fascinating. These tiny RNA molecules, typically only about 22 letters long, bind to messenger RNAs and interfere with their ability to be translated into protein, effectively turning down the volume on specific genes without switching them off entirely. Each microRNA can target dozens of different messenger RNAs, and each messenger RNA can be targeted by multiple microRNAs. The result is an intricate web of fine-tuned gene regulation that scientists are still in the early stages of understanding. Ribosomes, the molecular machines that translate messenger RNA into protein, are themselves largely made of RNA. The ribosomal RNA does most of the actual work of the ribosome, catalyzing the chemical reactions that link amino acids together. This was a surprising discovery because enzymes, biological catalysts, were thought to be exclusively proteins. RNA that can act as an enzyme is called a ribozyme, and its existence has given new life to an old idea: the RNA world hypothesis. The RNA world hypothesis proposes that life on Earth began not with DNA or proteins, but with RNA, a molecule that can both carry information, like DNA, and catalyze reactions, like proteins. In this view, DNA and proteins came later, evolving as more specialized and efficient tools, while RNA became the intermediary it is today. There is compelling evidence for this hypothesis, and it has become one of the leading frameworks for thinking about the origin of life. If life truly began with RNA, if the first molecules capable of self-replication were RNA, floating in some warm, mineral-rich pool or along the edges of a hydrothermal vent in the ancient ocean, then the RNA at work inside your cells right now is the descendant of the very first molecule that learned to copy itself. There is a continuity there that stretches back to the beginning of everything alive, and it hums quietly in every cell of every sleeping person in the world tonight. Please consider leaving a comment, sharing your favorite part of tonight's story. These conversations mean so much. And if you would like to explore more, our playlist has many more gentle evenings waiting for you. There is something almost musical about the way DNA works, not in a metaphorical sense, but structurally. The 4 letters of DNA are read in triplets, and each triplet codes for one of 20 amino acids. There are 64 possible triplets, 4 letters, arranged in groups of 3. 4 times 4 times 4 equals 64, but only 20 amino acids to encode, which means that most amino acids are encoded by more than one triplet. The genetic code is said to be degenerate, a slightly unfortunate word for something rather elegant. This redundancy has an interesting consequence. Because many codons can code for the same amino acid, some mutations that change a DNA letter do not change the amino acid that gets incorporated into the protein. These are called synonymous mutations, or silent mutations, changes at the DNA level that leave the protein unchanged. They are a kind of genetic cushion, absorbing mutations that might otherwise have consequences. Non-synonymous mutations are those that change the amino acid, and these can have effects ranging from completely harmless to profoundly significant, depending on where in the protein the change occurs and how the new amino acid differs from the original. The same chemical letter of the same gene, changed to a different letter, might have no effect in one position and a major effect in another. Context, in genetics as in language, matters enormously. Some mutations are not in the protein-coding parts of genes at all, but in the regions that control when and how much of the gene product is made. The promoter regions, the enhancers, the silencers. Mutations in regulatory regions can change the pattern of gene expression, the timing, the amount, the cell types in which a gene is active, without changing the protein itself. Many of the most important genetic differences between humans and chimpanzees are thought to lie in regulatory regions rather than in the genes themselves. Transposable elements, the mobile genetic sequences that make up a large fraction of the genome, have played a surprisingly important role in evolution. When they insert themselves into new locations in the genome, they can disrupt existing genes or create new regulatory connections. Over evolutionary time, some transposable elements have been domesticated, co-opted by the host genome to serve new functions. The immune system in particular appears to have borrowed mechanisms from ancient transposable elements at several points in its evolutionary history. The immune system's ability to generate an almost unlimited variety of different antibodies, proteins that can recognize and bind to virtually any foreign molecule, is itself a kind of genetic creativity. The genes that encode antibodies are assembled from a large collection of gene segments, shuffled and combined differently in each developing immune cell through a process called VDJ recombination. Each immune cell ends up with a unique antibody gene, a genome within the genome, edited in real time. Horizontal gene transfer, the movement of genes between organisms that are not in a parent-child relationship, is rare in animals but extremely common in bacteria. Bacteria can share genes with neighboring bacteria of entirely different species, passing useful capabilities, like antibiotic resistance, from one organism to another in what amounts to a kind of communal genome. This is one reason antibiotic resistance can spread so rapidly in bacterial populations. The full extent of horizontal gene transfer in the human genome is still being explored, but there is evidence that some human genes arrived not through the normal course of inheritance, but from ancient transfers, perhaps from bacteria, perhaps from ancient viral infections. The boundaries of the genome and the boundaries of species are somewhat more porous than they might initially appear. Telomerase is an enzyme that can extend telomeres, rebuilding the protective caps that shorten each time a cell divides. Most somatic cells, the non-reproductive cells of the body, have very little telomerase activity, which means their telomeres shorten over time, and their capacity to divide is limited. Stem cells and reproductive cells have higher telomerase activity, allowing them to divide more extensively. Cancer cells often reactivate telomerase, which is one of the ways they can divide indefinitely. The connection between telomere length, cellular aging, and overall health is a topic of active research. While shorter telomeres are associated with older biological age and certain health outcomes, the relationship is complex. Telomere length is influenced by genetics, lifestyle, and chance. It is not a simple dial that can be turned, but the gentle interest in understanding how cells age and how they might age more gracefully continues. There are moments in the history of science when a single discovery opens a door that had not even been noticed before. And beyond that door is a world so much larger than the one that came before it that it takes a generation to explore. The discovery of restriction enzymes in the 1970s was one such moment. Restriction enzymes are proteins produced by bacteria, again from bacterial immune systems, that cut DNA at specific sequences. Discovered by Werner Arber, Daniel Nathans, and Hamilton Smith, who shared the Nobel Prize in 1978, these molecular scissors made it possible for the first time to cut DNA in precise, predictable places. With restriction enzymes in hand, scientists could take pieces of DNA from one organism and insert them into another, the beginning of recombinant DNA technology, or genetic engineering. The first recombinant DNA organism was created in 1973. By 1982, insulin produced by genetically engineered bacteria was approved for use in medicine, the first recombinant protein therapy. Before this, insulin for diabetic patients had to be extracted from the pancreases of pigs and cattle. The supply was limited, and the insulin was slightly different from the human form. The techniques of recombinant DNA quickly proliferated into every corner of biology. Genes could be cloned, copied, and amplified, allowing scientists to work with specific sequences rather than having to sift through the entire genome every time. Gene libraries, collections of DNA fragments representing the full genome, could be made and stored and searched. Specific proteins could be produced in large quantities by inserting the relevant gene into bacteria or yeast and letting them grow. The polymerase chain reaction, PCR, was invented by Kary Mullis in 1983, and it transformed biology as thoroughly as restriction enzymes had a decade before. PCR allows a specific short stretch of DNA to be amplified billions of times in just a few hours, starting from vanishingly small amounts of starting material. A single hair, a tiny drop of dried blood, a fragment of ancient bone. All can provide enough DNA for analysis if PCR is used to amplify the regions of interest. PCR became familiar to many people during the COVID-19 pandemic, when PCR tests were used to detect the genetic material of the SARS-CoV-2 virus in patient samples. The same fundamental technique that Mullis developed in the 1980s, scaled and automated, became one of the primary tools of a global public health response. Gel electrophoresis, another foundational technique, allows DNA fragments of different sizes to be separated based on how far they travel through a gel when an electric current is applied. Shorter fragments move further, longer fragments less so. The result is a pattern of bands that can be read and compared. It is the technique that gave us the iconic DNA fingerprint images, ladders of bands that represent the pattern of a person's DNA at multiple locations in the genome. DNA microarrays, sometimes called gene chips, allowed scientists to measure the activity of thousands of genes simultaneously. A microarray is a small surface, often glass, dotted with thousands of short DNA sequences. When you apply a sample of RNA from a cell or tissue, the RNA sticks to its complementary sequences on the array, and the pattern of sticking reveals which genes were active in the original sample. This technology made it possible, for the first time, to take a snapshot of gene expression across the entire genome at once. These technologies have not just been tools for understanding biology. They have also been tools for understanding ourselves. Genealogical databases built from millions of DNA tests have made it possible to identify previously unknown relatives, to connect with distant cousins, to trace family histories across continents. They have also allowed investigators working through public genealogical databases to identify suspects in old crimes by matching crime scene DNA to relatives who had submitted their DNA for ancestry testing. The ethical conversations these technologies have opened about privacy, about consent, about who owns your genetic information and what can be done with it, are ongoing and important. Science does not arrive neatly packaged with all its ethical implications already resolved. It arrives as a possibility, and the work of deciding how to use it wisely belongs to all of us. Let yourself rest now with these large and quiet thoughts. You do not need to resolve them tonight. Just hold them lightly, the way you might hold a smooth stone in a warm hand, present, real, and asking nothing of you. One of the gentler surprises in genetics is how much we do not know. The human genome has been sequenced completely now, but reading the letters is not the same as understanding them. Scientists estimate that the functions of a large fraction of the genome are still unclear, still being worked out, still surprising researchers with new connections and new capabilities. The ENCODE project, the Encyclopedia of DNA Elements, was a large international collaboration that began after the Human Genome Project and aimed to identify all the functional elements in the human genome. Its results, published in 2012 and updated several times since, suggested that much more of the genome is functionally active than previously thought. Though exactly what functional means in this context and how to define it has been an ongoing conversation in the field. What is becoming increasingly clear is that the genome is not a simple instruction manual with clearly labeled sections. It is more like an ancient and extraordinarily complex document, written in a language only partially understood, with layers of annotation and cross-reference that interact in ways still being discovered. New functional elements keep being identified. New regulatory relationships keep being uncovered. The more carefully scientists look, the more there is to see. One of the most surprising recent discoveries has been the extent to which the three-dimensional folding of the genome, how different regions of DNA come close to each other in the nucleus, affects gene regulation. Distant regions of DNA can interact with each other when they are folded into spatial proximity, allowing enhancers, regulatory elements that boost gene expression, to communicate with promoters, the on switches of genes, even when the two are separated by millions of letters of DNA sequence. The linear map of the genome, it turns out, is only part of the story. Long non-coding RNAs, RNA molecules transcribed from the genome that do not code for protein and are longer than 200 letters, have emerged as a major class of regulatory molecules whose full range of functions is still being worked out. There are tens of thousands of them in the human genome. Some have been linked to development, to disease, to the establishment of chromatin architecture, the three-dimensional organization of DNA in the nucleus. Others remain enigmatic, their functions yet to be determined. The study of the microbiome, the community of bacteria, fungi, and other microorganisms that live in and on the human body has added yet another layer to the understanding of how genetics influences health. The microbiome has its own genome. Collectively, the microbial genomes associated with a human body contain vastly more genes than the human genome itself, and it interacts with the human genome in ways that are still being mapped. The gut microbiome, in particular, appears to influence everything from digestion and immune function to mood and cognition. Epigenetic clocks, mathematical models trained on patterns of DNA methylation that change predictably with age, can estimate a person's biological age from a blood or tissue sample with surprising accuracy. Biological age can differ from chronological age, and researchers are investigating what factors cause the two to diverge, and whether biological age can be slowed or reversed in specific contexts. The search for so-called longevity genes, genetic variants associated with living to very old age in good health, has identified some candidates, but no single gene or set of genes has emerged as a simple answer. Long life seems to involve a complex interaction of genetic factors, lifestyle, environment, and chance, the same intricate interplay that underlies most complex human traits. Single-cell genomics, the ability to sequence the genome or measure gene expression in individual cells rather than averaging across millions, has revolutionized the understanding of cell types and cell states. Every tissue in the body turns out to contain far more diversity than was visible by looking at cells under a microscope. The brain alone is estimated to contain hundreds of distinct cell types, each with its own characteristic pattern of gene expression, its own particular identity written in which sections of the shared genome it chooses to read. This is one of the most extraordinary things about DNA. It is not just one text. It is a library from which each cell selects its own reading list, and those reading lists together produce the astonishing, humming, quietly miraculous complexity of a living body. We are near the end of our journey tonight, And if you have found your way here through soft attention and gentle drowsiness, that is exactly as it should be. Please leave a like if these stories have given you a peaceful night. And there is always more waiting for you in the playlist. As we drift toward the quiet end of tonight, let the thoughts settle like dust in a ray of afternoon light, slowly, without effort, finding their resting place. Your DNA has been traveling a very long road to be here with you tonight. Every ancestor who survived a winter, who found food in a difficult season, who recovered from illness or injury or hardship, each of them passed their DNA forward, a baton carried across unimaginable distances of time until it arrived here inside you in this moment. The letters of your genome were assembled over billions of years, some of them so ancient that they predate the emergence of complex life, so conserved that they have been faithfully copied through every generation of life on Earth since before animals existed. And some of them are particular to you, to your family, to the specific combination of people and places and moments that led to your existence. There is something that feels like a kind of belonging in this. Not a belonging that requires anything of you, not a belonging you have to earn or maintain, but simply the quiet fact of being part of an unbroken chain of life that has never once faltered, never once failed to carry its message forward, not in 4 billion years. Your cells are working right now, gently, tirelessly, without asking for your attention. DNA is being read, RNA is being made, proteins are folding into their shapes, energy is being produced, old cells are being quietly retired, and new ones are taking their place. All of this happens in the space of a fraction of a blink, and it happens without you needing to do anything at all. You are, in the most literal sense, an expression of a very long sentence, one that began almost at the beginning of life on Earth and has been writing itself forward ever since, finding new ways to say what it has always been trying to say, that life continues, that warmth is real, that the morning comes. The secrets of DNA are not locked away in distant laboratories or hidden in technical language. They are here in your body, happening right now, in the warmth of your breath, the steady rhythm of your heart, the gentle weight of your limbs against the surface beneath you. You are made of the same substance that makes everything alive, and that substance has been finding its way across time for longer than the mind can truly hold. There is nothing you need to do with any of this tonight. You do not need to remember it, or understand it fully, or do anything with it at all. It is simply here, the way the stars are simply there, whether you look at them or not. Let your breathing slow a little more. Feel the quiet around you. The room is still. The night is soft. Your body knows exactly what to do. If tonight's story has been a gentle companion on the way to sleep, please leave a comment, leave a like. It helps us reach more people who might need a soft place to rest. Our playlist is always there, waiting like a lamp in a window for whenever you need it. Close your eyes now, if you haven't already. Let the words dissolve into warmth. The story does not end. It simply continues quietly, in every cell of every living thing, on and on through the night and into whatever morning comes next. You are safe. You are warm. You are made of something ancient and patient and quietly extraordinary. Sleep well. Sleepy Science Channel. Gentle stories for curious minds at rest. #DNASecrets. parts 16 to 19. There is a particular kind of wonder that belongs to the very small, not the wonder of mountains or oceans wide open, available to anyone who turns their face toward them, but the wonder of the hidden, the wonder of what is there, whether you look or not, going about its quiet business in the dark interior of things. DNA lives in that kind of wonder. It does not need to be observed to do what it does. It was working long before anyone knew it existed, and it will continue long after tonight has folded into morning. One of the quieter revolutions in our understanding of DNA has been the realization that the genome is not read the same way in every moment of a cell's life. Cells respond to their environment, to signals from neighboring cells, to changes in temperature, to the availability of nutrients, to the rhythms of day and night, by adjusting which genes are active and which are not. The genome is not a fixed broadcast, but something more like a conversation, constantly responsive, constantly adapting. Circadian rhythms, the roughly 24-hour biological cycles that govern sleep and wakefulness, hormone release, body temperature, and many other processes, are written, in part, in the genome. A set of genes called clock genes interact with each other in a feedback loop. They produce proteins that accumulate over several hours, then switch off the genes that made them, then gradually break down, allowing the genes to become active again. This molecular clock ticks inside nearly every cell in your body, keeping time with the rotation of the Earth. The master clock for the body's circadian rhythms is located in a small region of the brain called the suprachiasmatic nucleus, a cluster of only about 20,000 neurons sitting just above the point where the optic nerves cross. These cells receive light information from the eyes and use it to synchronize the molecular clocks in cells throughout the body. Light filtered through your eyes sets the pace of gene expression in cells from your liver to your skin. Even individual genes can show circadian variation in their expression, rising and falling over the course of a day in predictable rhythms. Studies have found that somewhere between 10 and 80% of the genome, depending on the tissue and the species studied, shows some degree of circadian oscillation in gene expression. The genome breathes, in a sense, with the rhythm of the day. Sleep itself, the state you may be drifting toward right now, is deeply intertwined with gene expression. During sleep, certain genes involved in cellular repair, memory consolidation, and immune function become more active. The brain clears metabolic waste products that accumulate during waking hours. Proteins damaged by the work of the day are tagged for replacement. Sleep is not the absence of biology. It is a different mode of it, one that the genome has been refined to support across hundreds of millions of years of evolution. Dreams, which occur primarily during a phase of sleep called REM, rapid eye movement, sleep, are associated with a particular pattern of brain activity. The neural circuits that process emotion and memory are especially active during REM sleep, while the prefrontal cortex, associated with rational thought and self-monitoring, is relatively quieter. Whether dreams serve a specific function related to memory consolidation, emotional processing, or simply reflect the background activity of a resting brain is still an open question, debated warmly and without resolution in sleep research. The genes involved in sleep regulation are ancient. Many of the core clock genes in humans have counterparts in fruit flies, animals that separated from the human lineage more than 500 million years ago, and those counterparts perform similar functions. The molecular clock is one of the most conserved biological systems known, suggesting that timed rhythmic gene expression was an important adaptation very early in the history of life. Jet lag, the disorientation that comes from traveling rapidly across time zones, is, at its core, a disruption of circadian gene expression. The body's internal molecular clocks fall out of synchrony with the external light-dark cycle of the new location, and the various clocks in different tissues resynchronize at different rates. The liver's clock might take several days to adjust, while the brain's master clock adjusts more quickly. And in the meantime, the body's internal signals are out of step with each other, producing the familiar fog and fatigue. Social jet lag, a term coined by sleep researchers to describe the misalignment between a person's biological clock and their social schedule, affects many people who keep different hours on weekends than on weekdays. This chronic, mild disruption of circadian rhythms has been associated in studies with various health outcomes, and it is one of the quieter ways in which the modern world's scheduling can work against the body's ancient rhythms. Melatonin, the hormone most commonly associated with sleep, is produced by the pineal gland in the brain, and its production is directly regulated by light input to the suprachiasmatic nucleus. In the absence of bright light, melatonin production rises in the evening, signaling to the body that night is approaching. The genes that encode the enzymes responsible for melatonin synthesis are among the clock-regulated genes. Their expression rises and falls with the time of day, orchestrated by the molecular clock. There is something peaceful in knowing that your body has been keeping time since long before clocks were invented, that the rhythm of sleeping and waking, of rest and activity, is written into the deep structure of your biology, shared with creatures as different from you as fungi and sea anemones. You are not outside the rhythm of the natural world. You are part of it, breathing in time with it, even here, even now. If you are following along from a place of growing drowsiness, that is exactly right. Leave a like on this video if it is becoming part of your sleep routine. And our playlist is always there, one gentle tap away whenever you need it. The microRNA molecules that regulate gene expression during sleep are a field of active and rather beautiful research. These tiny regulatory RNA molecules, remember, only about 22 letters long, shift in their abundance and activity between sleep and wakefulness, fine-tuning the expression of dozens of genes simultaneously. They are like a gentle dimmer switch for the genome, adjusting the light rather than simply turning it on or off. The relationship between DNA and memory is one of the most quietly astonishing areas of current research. Long-term memories, the ones that persist across years and decades, require changes in the strength of connections between neurons, and those changes require the synthesis of new proteins, which requires gene expression. A memory at the molecular level is partly written in the pattern of which genes in which neurons were activated at the moment the memory was formed, and in the structural changes that followed. There is even evidence that the formation of strong memories involves transient changes to the physical structure of DNA in neurons. Small, reversible breaks that may help remodel the chromatin around memory-related genes, making them more accessible to be read. The idea that remembering something involves, at a molecular level, a kind of controlled and repairable disruption to DNA in specific neurons is striking, and researchers are still working to fully understand what it means. The genome, in other words, is not just a blueprint. It is an active participant in everything the body does, in sleep, in waking, in memory, in forgetting, in the quiet maintenance of the self across time. It listens to the world through the signals that reach it, and it responds. It is less like a book than like a conversation, ongoing, contextual, never quite the same twice. And that conversation is happening right now in every cell of your body. As the room grows quieter and your breathing deepens and the thoughts of the day grow softer at their edges, nothing is required of you. The conversation continues without you needing to participate consciously. You can simply rest. Among the many things that make DNA remarkable is its relationship with time. Not just evolutionary time, the long, slow river of species changing across millions of years, but the more intimate time of a single life. From the moment of fertilization to the last quiet breath, DNA is both the constant thread and the site of continuous change, a river that is always the same river and never the same water. Development, the process by which a single fertilized cell becomes a complete organism, is one of the most profound expressions of what DNA can do. The fertilized egg contains no special structures, no obvious organization. And yet, from that one cell, in a process governed by the interactions of thousands of genes and the physical forces they create, a body assembles itself with extraordinary reliability. The same process, starting from essentially the same conditions, produces a recognizable human being every time. The genes that orchestrate the basic body plan, that establish which end will be the head and which the tail, which side will become the back and which the belly, how many segments the body will have, and what structures will form in each, belong to an ancient family called the Hox genes. Hox genes are found in nearly all animals, from flatworms to humans, and they are arranged along the chromosome in the same order as the body parts they control. The genes that govern the head are at one end, and those that govern the tail are at the other. This collinearity between gene order and body axis has been conserved for more than 500 million years. Disruptions to Hox gene expression produce striking and strange results in model organisms. Flies with legs growing where antennae should be, or extra pairs of wings. These homeotic mutations, as they are called, were among the first clear demonstrations that single genes could have sweeping effects on body organization, and they opened the door to the field of evolutionary developmental biology, Evo-Devo, which explores how changes in developmental genes drive the evolution of new body forms. The similarities between the developmental programs of very different animals are one of the more quietly wonderful facts in all of biology. The genes that direct the formation of the eye in a fruit fly are so similar to those that direct eye formation in humans that, remarkably, a human eye-forming gene inserted into a fly can direct the formation of a fly eye. The deep logic of how to make an eye is shared, even though the eyes themselves are quite different in structure. Evolution is extraordinarily conservative about its core tools. Stem cells, cells that retain the ability to divide and differentiate into many different cell types, hold a particular fascination in biology because they represent a kind of developmental potential, a not-yet-ness that most cells lose as they mature. Embryonic stem cells, found in the very early embryo, can give rise to any cell type in the body. Adult stem cells, found in various tissues throughout life, are more restricted. They typically give rise to the cell types of the tissue in which they reside. In 2006, the Japanese researcher Shinya Yamanaka discovered that ordinary adult cells could be reprogrammed back to a stem cell-like state by introducing just 4 specific genes encoding transcription factors, proteins that regulate the activity of other genes. The resulting cells were called induced pluripotent stem cells, or iPSCs, and Yamanaka received the Nobel Prize for the discovery in 2012. The findings suggested that cell identity is not permanently fixed, that the epigenetic modifications that define a mature cell's identity can, under the right conditions, be largely reversed. Transcription factors, the proteins that bind to specific INA sequences and either activate or repress nearby genes, are among the most powerful regulators of gene expression. A single transcription factor can control hundreds of target genes simultaneously, and combinations of transcription factors working together can specify complex cell identities. The idea that just a handful of these proteins can reprogram an adult cell's entire gene expression profile, and thereby its identity, speaks to the combinatorial power of gene regulation. The timing of gene expression during development is as important as the identity of the genes themselves. Genes that are expressed at the wrong moment, too early, too late, can have profound effects on the resulting organism, even if the gene sequence itself is normal. This temporal dimension of gene regulation adds another layer of complexity to the genome's operation. Not just which genes are on, but when. Imprinting is one of the more counterintuitive aspects of gene regulation. In most cases, both copies of a gene, one inherited from each parent, are available to be expressed. But for a specific subset of genes, one copy is silenced based on which parent it came from. This silencing is established by epigenetic marks set in the egg or sperm, and it persists through development. The result is that, for imprinted genes, only the maternal or only the paternal copy is active, and disruptions to imprinting can have significant developmental consequences. The evolutionary explanation for imprinting is a matter of ongoing scientific discussion, but one influential theory frames it in terms of a kind of genetic conflict between the interests of maternal and paternal genes, a quiet molecular negotiation written into the epigenome, playing out in the developing embryo without any awareness on anyone's part. X inactivation is another form of selective gene silencing, specific to people with 2 X chromosomes. Since 2 active copies of every X-linked gene would produce twice the protein compared to cells with only 1 X chromosome in each cell is largely silenced, a process that occurs early in development and produces the characteristic cellular mosaicism of people with XX chromosomes. The silenced X chromosome condenses into a compact structure called a bar body, visible under a microscope as a small, dense spot in the nucleus. Which X is silenced in each cell, the maternal or the paternal, is largely random, meaning that the body of a person with two X chromosomes is a mosaic of two populations of cells, each expressing a different set of X-linked genes. In cats, this mosaic is visible in the coat. The tortoiseshell pattern, with its irregular patches of orange and black, is the visible expression of X inactivation in the pigment cells of the skin. Perhaps that is a gentle image to carry towards sleep. A sleeping tortoiseshell cat, its coat a visible map of its cellular mosaic, each patch of color a quiet record of which X chromosome was silenced in that particular cluster of cells long ago in the early days of its development. DNA making itself visible on the surface of things in patterns that are beautiful without trying to be. Please leave a comment if you enjoyed tonight's story. Tell us which fact surprised you most. And remember, there is always more in the playlist for the nights when sleep day takes a little longer to arrive. The proteins that DNA encodes are only the beginning of the story. What proteins do, how they fold, where they go, who they interact with, how they are modified after they are made, determines the actual biology of the cell, and that biology is vastly more complex than the genome alone can explain. Protein folding is one of the great puzzles of molecular biology. The sequence of amino acids in a protein determines its three-dimensional shape, but predicting that shape from the sequence alone, a problem known as the protein folding problem, resisted solution for decades. The shape of a protein determines its function, so understanding folding is essential to understanding what any given protein does. In 2020, an artificial intelligence system called AlphaFold, developed by DeepMind, produced remarkably accurate predictions of protein structures from amino acid sequences, solving, in large part, a problem that had seemed intractable for 50 years. The developers released a database of predicted structures for nearly all known proteins, freely available to researchers around the world. It was one of the most celebrated achievements of AI in science, and it has already begun to accelerate research in areas from drug discovery to the understanding of disease mechanisms. Proteins rarely work alone. Most cellular processes involve large complexes of multiple proteins working together, fitting together in precise three-dimensional arrangements, like the interlocking parts of a very small and very intricate machine. The study of these interactions, the interactomy, is one of the frontiers of systems biology, which tries to understand biology not as a collection of individual components, but as a network of interactions. Post-translational modifications, chemical changes made to proteins after they are synthesized, add yet another layer of complexity. Proteins can be phosphorylated, a phosphate group added, ubiquitinated, tagged for destruction, acetylated, glycosylated, and modified in dozens of other ways, each change affecting the protein's activity, location, stability, or interactions. The number of distinct protein species in a cell is vastly larger than the number of genes, because each protein can exist in many differently modified forms. Chaperone proteins are molecules whose job is to help other proteins fold correctly, to catch them before they misfold, to unfold them if they have gone wrong, and to give them another chance to find their correct shape. The need for chaperones reflects the difficulty of the folding problem from the protein's perspective. The space of possible shapes is enormous, and finding the correct one reliably requires help. Cells under stress, heat for instance, produce extra chaperones, which is why these proteins were originally discovered as heat shock proteins. When proteins do misfold and aggregate, clumping together into insoluble masses, the results can be severe. Several neurodegenerative diseases, including Alzheimer's disease and Parkinson's disease, are associated with the accumulation of misfolded protein aggregates in the brain. The connection between these aggregates and the disease process is complex and still under investigation. But understanding protein misfolding is one of the central goals of research into these conditions. The proteasome is the cell's protein recycling system, a large barrel-shaped complex that degrades proteins tagged for destruction by chains of ubiquitin molecules. The proteasome breaks proteins down into their component amino acids, which can then be reused to make new proteins. This constant recycling of proteins is essential to cellular health, allowing the cell to remove damaged or unneeded proteins and respond to changing circumstances by rapidly adjusting the protein composition of the cell. Secreted proteins, those that are made inside the cell but then released to act outside it, include hormones, growth factors, antibodies, and many of the proteins involved in communication between cells. These proteins travel through the bloodstream or through the spaces between cells, binding to receptors on distant cells and triggering changes in gene expression. In this way, the genome of one cell can influence the behavior of cells far away, an extraordinary reach for something that lives inside a nucleus too small to see. The insulin molecule, which regulates blood sugar, is one of the most studied secreted proteins. It is encoded by a single gene on chromosome 11, produced in the beta cells of the pancreas, processed through several steps of modification, stored in vesicles inside the cell, and then released in response to rising blood sugar levels. The entire cascade, from gene expression to protein synthesis, to modification, to storage, to release, to receptor binding, to downstream gene regulation in target cells, is a marvel of coordinated molecular action happening quietly in your body after every meal. Signal transduction, the process by which a signal from outside the cell is converted into a change in gene expression inside the nucleus, typically involves a chain of molecular events, each step amplifying or modulating the signal before passing it on. A single signaling molecule binding to a receptor on the cell surface can, through a cascade of molecular interactions, activate or repress hundreds of genes in the nucleus. The amplification is remarkable. A whisper at the cell surface can become a roar in the nucleus, or a roar at the surface can be carefully dampened to a murmur. Receptors themselves are proteins, encoded by genes, and the diversity of receptors a cell expresses determines what signals it can receive, which is another way in which gene expression shapes cellular identity and behavior. A cell that does not express the receptor for a particular hormone cannot respond to that hormone, no matter how much of it is present in the surrounding environment. The genome, in this sense, determines not just what a cell is, but what it can hear. The olfactory system, the sense of smell, is, genetically speaking, one of the most extravagant investments the mammalian genome makes. Humans devote somewhere around 400 active olfactory receptor genes to detecting smells, and in mice, the number is even higher, closer to 1,000. Each olfactory receptor neuron in the nose expresses only one olfactory receptor gene, chosen from the full set through a mechanism that is still not fully understood, and sends its signal to a specific location in the brain. The result is a combinatorial code of receptor activations that allows the brain to distinguish thousands of different odors. The scent of rain on dry earth, petrichor, the smell of bread baking, the particular warm smell of a sleeping person's hair. All of these experiences begin with molecules binding to olfactory receptor proteins encoded by genes in the genome, triggering electrical signals that travel to the brain and are decoded into perception. Smell is, in this sense, a translation. The chemical world outside converted letter by molecular letter into experience. Rest now in the quiet of that translation. The world is always writing to you in molecules you cannot see, and your genome is always reading the message. Tonight, the message is, be still, rest. The work is done for now. As the night deepens and the thoughts grow quieter, let the last few stories come to you like light through water. Present, gentle, not requiring anything. The story of DNA is, among other things, a story of information. Before life, there was chemistry, molecules forming and breaking apart, reactions proceeding and reversing, energy flowing from place to place. At some point, in some warm, mineral-rich environment that we cannot yet precisely identify, a molecule appeared that could do something new, copy itself. That moment, whenever and wherever it happened, was the beginning of information in biology. Everything since has been an elaboration on that first copying. Information, in the technical sense used by physicists and mathematicians, is about the reduction of uncertainty. A genome carries information because knowing its sequence tells you things about what proteins will be made, about what the organism will look like, about how it will develop and behave. The amount of information in a human genome is vast, measured in gigabytes of digital storage. And yet, it is compressed, folded, regulated, and read with a sophistication that no human-designed information system yet approaches. The field of DNA data storage explores using DNA itself as a medium for storing digital information. Because DNA can encode information in its sequence of 4 letters, with each letter representing 2 bits of binary data. DNA is an extraordinarily dense storage medium. A single gram of DNA, theoretically, could hold about 200 petabytes of data, roughly 100 times the total content of the entire internet in the early 2020s. And DNA, under the right conditions, can remain readable for hundreds of thousands of years. Researchers have demonstrated the storage and retrieval of text, images, and even video in synthetic DNA sequences. Synthetic biology, the design and construction of new biological systems, including new DNA sequences encoding proteins or regulatory circuits that do not exist in nature, has grown rapidly as a field in the past 2 decades. Synthetic biologists have built genetic circuits that function like electronic logic gates, creating cells that can perform computations, detect specific chemicals, and produce useful molecules in response. The first complete synthesis of a bacterial genome, approximately 1 million letters of DNA assembled from scratch, was reported in 2010 by Craig Venter's team. The resulting synthetic genome, when transplanted into a recipient cell, directed the cell's behavior, demonstrating that a genome assembled letter by letter in a laboratory could function as a living genome. The prospect of designing organisms from the ground up, of writing new genomes as one might write code, raises profound questions about the relationship between living and made, between natural and artificial, between the discovered and the invented. Biology has always involved redesigning living things through selective breeding, which is itself a form of genome editing, just much slower and less precise. The new technologies compress that timeline enormously and expand the scope of what is possible. And with that expansion comes the need for careful thought about how the possibilities are used. There are researchers working on the possibility of resurrecting extinct species, or at least of creating living animals that carry some of the genes of extinct relatives, using ancient DNA and modern gene editing. The woolly mammoth is perhaps the most discussed candidate. Its genome has been sequenced from permafrost specimens, and some of the genes that adapted it to cold environments have been identified. Whether full resurrection is biologically possible or desirable or wise is a question that biologists, ethicists, and ecologists are actively discussing. The bacterium Deinococcus radiodurans is sometimes called the world's most radiation-resistant organism, and it holds an unusual record in the context of DNA. It can survive radiation doses that shatter its genome into hundreds of fragments, and then reassemble those fragments correctly, stitching its DNA back together from the pieces. Its DNA repair machinery is so efficient and redundant that it can reconstruct a shattered genome with remarkable fidelity. It is, in a sense, the most resilient keeper of genetic information known. Tardigrades, microscopic animals often called water bears, about 0.5 millimeter long, can survive conditions that would destroy virtually any other animal. Extreme dehydration, the vacuum of space, intense radiation, temperatures from near absolute zero to well above boiling. When dried out, they enter a state called cryptobiosis, in which all metabolism stops and they become essentially inert. And they can remain in this state for decades, resuming active life when water is reintroduced. Their genomes encode a range of protective proteins that defend against damage during these extremes, and studying them has revealed new insights into how living systems can protect their DNA under stress. DNA itself, removed from all cellular context, is remarkably stable under the right conditions. The structure of the double helix, the hydrogen bonds between complementary base pairs, the stacking interactions between adjacent base pairs, gives it a physical stability that allows it to persist for thousands of years in cold, dry environments. The sequencing of DNA from ancient remains, from woolly mammoths, from Neanderthals, from ancient humans, has been possible precisely because of this inherent stability. The sequence of the human genome has been described as the book of life, and the metaphor is both apt and incomplete. It is apt because the genome contains the instructions for building and operating a human being, and those instructions are written in a precise, readable sequence of chemical letters. It is incomplete because the genome, unlike a book, does not have meaning on its own. It has meaning only in context, only in the presence of the cellular machinery that reads it, only in interaction with an environment, only as part of a living system embedded in a world. A genome in isolation is just chemistry. A genome inside a living cell, communicating with other cells, embedded in a body, nested in an environment, connected through time to all the genomes that preceded it. That is life. And life, whatever else it is something that reads its own instructions and acts on them, something that maintains itself and reproduces itself and changes over time, something that has been finding its way forward through every difficulty and every abundance since the very first molecule learned to make a copy of itself in some warm and ancient water. You are part of that story, not a spectator, not an observer, but a living chapter, written in the same language as everything else alive, carrying the same ancient letters, reading them in your own particular way, your own unrepeatable combination of inherited text and lived experience, and the specific, irreducible fact of being you. There is nothing more you need to do tonight. The story continues by itself, quietly, in the dark, in every cell of your body, in every sleeping creature in the world, outside your window, in the soil and the sea, and the slow, cold depths of the night. Let your eyes be closed, if they are not already. Let your breath find its own slow rhythm. Let the thoughts dissolve, one by one, like words spoken softly in an empty room. You have traveled a long way tonight, through the structure of the double helix, through the copying and repair of DNA, through the vast history written in your genome, through the languages of epigenetics and development and evolution and time. And all of it has brought you here to this quiet moment, at the edge of sleep. Thank you for spending this time with us. If these stories have given you rest, please leave a comment, leave a like. It is how we know these gentle hours are finding the people who need them. And our playlist will always be here, a soft collection of nights just like this one, waiting for whenever the world feels too loud and sleep feels too far away. Close your eyes. You are safe. You are warm. The double helix turns slowly in the dark, patient and faithful, carrying everything forward into morning. Sleep well. Sleepy Science Channel. Gentle stories for curious minds at rest.